Today we're going to be talking about the process of transcription of DNA into RNA in prokaryotic cells. This is the general model for transcription in prokaryotes. We have our double-stranded DNA up top in black, and we have our single-stranded RNA in red on the bottom. Now let's start at the beginning. It all starts with a thing called the holoenzyme. Now what the holoenzyme is it's a combination of the RNA polymerase, which is all the factors that are needed to code RNA, and the sigma factor, which bonds to the promoter region, which is seen up here. Now in this promoter region, we have two major sections. We have -10 and -35. And these are counted to from our +1 nucleotide, which is the first nucleotide in the coding region. There's no zero nucleotide, it just goes from +1 to -1. This -10 region over here is what we call the Tata box, and that's because it has high amounts of T's and A's in its sequence, and we also have the -35 region back here, which is also pretty standard across organisms. Now these two regions is what our sigma factor finds when it's looking to code RNA. Once our sigma factor bonds here, it wants to stay here, and it lets the RNA polymerase go on to code the RNA coding region. This region starts at, of course, our +1 and ends with a terminator, which stops the RNA coding. Now let's take a look at our two strands of DNA. Up top, we have the coding strand, and on the bottom, we have our template strand with our DNA, with our RNA. The template strand is always closer to the RNA, and it is what the RNA is built off of. It's the template, which is how it gets its name. The other strand, the coding strand, holds the exact sequence of the RNA minus the uracil. And this is what really determines what our RNA is going to be, while the template strand is just a mere messenger. Now it's also important to notice that RNA grows in the 3' direction, which runs anti-parallel to our template strand and parallel to our coding strand. There are two types of termination. We have row-independent and row-dependent. Row-independent uses a hairpin loop consisting of inverted repeats to pull on the RNA strand away from the DNA and causes separation. And then there's row-dependent, which uses a row subunit, which travels up the RNA and uses helicase activity to separate it from the DNA by unwinding it. So what if we had no -10 region? If there was no -10 region in a piece of DNA that coded for an enzyme that metabolized sugar, what would happen? So if this happened, that enzyme would not be created, which means that sugar would not be metabolized fast enough by the cell, and that cell would die because it doesn't receive enough energy. And here are the works cited.